The human organism is an open thermodynamic system, maintaining homeostatic stability through the continuous exchange of energy with its environment. While usually associated with vision or heat, the electromagnetic spectrum actually serves as a primary driver of cellular respiration and genomic expression. To understand modern metabolic disease, we must map how specific wavelengths of light dictate either metabolic coherence or oxidative toxicity. The infinite fractal descent model describes this process, where electromagnetic energy cascades across multiple biological scales, from photons to behavior. This model suggests that shifts at the quantum scale propagate upward, eventually manifesting as changes in systemic physiological states. Light functions as a measurable metabolic nutrient, and its biological translation is governed by the predictable constraints of physical laws. Short-wavelength radiation, spanning 100 to 490 nanometers, possesses high photonic energy. This energy induces direct photochemical alterations in the epidermis and the retina. In the skin, ultraviolet B triggers the synthesis of vitamin D and the transcription of the POMC gene. This reaction releases beta endorphins and sex steroid hormones into the system. These endogenous opioids produce an analgesic effect, driving the evolutionary addictive behavior often described as solar-seeking. Ultraviolet A operates through a different mechanism, facilitating the non-enzymatic release of nitric oxide from photolabile stores within the skin. This nitric oxide acts as a vasodilator. Clinical data shows that whole-body UVA exposure of 20 joules per square centimeter reduces systemic blood pressure within 30 minutes. Blue light serves as the primary zeitgeber for human circadian rhythms, interacting with melanopsin-containing cells in the retina to suppress melatonin. Daytime blue light optimizes metabolic rate, yet modern artificial light at night creates an evolutionary mismatch that disrupts these internal timing mechanisms. UK Biobank data involving over 84,000 participants links brighter nightlight exposure to a 1.53 hazard ratio for incident type 2 diabetes. Short-wavelength light functions as the organism's environmental sensor, immediately translating superficial radiation into systemic hormonal and vascular commands. Long-wavelength radiation, from 600 to 1,000 nanometers, penetrates deeply into biological tissues, reaching the muscle layer without the high-energy DNA damage associated with UV light. At this scale, cytochrome C oxidase, the terminal enzyme in the mitochondrial electron transport chain, serves as the primary receptor for red and near-infrared photons. Absorption of 670 nanometer photons provides the energy to photodissociate inhibitory nitric oxide from the enzyme's catalytic centers, removing the physical brake on cellular respiration. This unbraking increases ATP production and causes the cell to pull more glucose from the bloodstream to fuel the accelerated metabolic demand. A 2024 study by Pauner and Jeffrey provides the clinical validation for the systemic glucose modulation. A single 15-minute exposure to 670 nanometer red light reduced integrated postprandial blood glucose elevation by 27.7% following a glucose challenge. Near-infrared light reaches even deeper into the tissue, polarizing macrophages toward a reparative phenotype to support angiogenesis and muscle recovery. Red and near-infrared photons function as physical fuel. The mitochondria consume this energy to accelerate deep tissue repair and stabilize glucose clearance. The biological utility of light is restricted by thermodynamic limits defined by the specific dose or fluence of radiation. This is the Arndt-Scholz law, a nonlinear, biphasic response where weak stimuli accelerate physiologic activity while excessive stimuli inhibit it. Within the Goldilocks zone, photon absorption triggers protective transcription and promotes homeostasis. When the optimal dose is exceeded, the curve descends into the negative axis, inhibiting biological function. At this threshold, the system hits a Ross bottleneck, leaking electrons and creating excessive oxidative stress. This toxicity manifests as mitochondrial DNA damage and the thermal degradation of collagen, marking the boundary where energy becomes destructive. Dose determines the outcome. The same wavelength can provide either biological coherence or oxidative toxicity, depending on the thermodynamic threshold. This biphasic relationship becomes critical when evaluating modern environments, where artificial lighting mimics sunlight visually but not spectrally. Our biology evolved with spectral buffering. Sunlight's yellow line shows high-energy blue spikes balanced by reparative red wavelengths. Modern LEDs lack this, providing an isolated blue stimulant spike at 450 nanometers. Past 650 nanometers, the spectrum flatlines, offering zero red energy for cellular repair. This environmental condition is known as the red starvation hypothesis. Without these near-infrared photons, cytochrome C oxidase remains inhibited, reducing our capacity for mitochondrial respiration and cellular repair. Following the infinite fractal descent model, this chronic lack of quantum-scale red photons eventually cascades into systemic metabolic dysfunction. This spectral mismatch is a contributing factor to the global rise in insulin resistance and chronic oxidative stress. Light operates as a structural component of human metabolism, acting as a metabolic nutrient rather than a source of illumination. Maintaining this spectral balance, aligning specific wavelengths with their appropriate doses, is a central requirement for metabolic health in indoor environments.